So, over the years, humans have altered native habitats for their own benefits for residential, industrial, and agriculture purposes. When the native land is altered, it can have a negative effect on the native species and their habitats. It can cause habitat isolation, habitat fragmentation, and habitat loss. If there is a creek or a stream running through the native habitat that is about to be altered, it can impact the water quality of the stream and the aquatic species habitats. And this can affect the fish distribution and especially the stream fishes. Alterations can impose stressors on aquatic species. There's been previous studies done on other species that showed an increase in temperature can disturb the growth. Also, the lower dissolved oxygen level can increase the stress level of juvenile fish, and pollution can alter pH, which can harm the aquatic species. Now, whenever a fish or other animals get stressed, they release cortisol through the process of the hypothalamus pituitary entorhinal axis. Now, if it's an acute stress, that's okay. It's actually beneficial for the animal. But if it's a chronic stressor, this can be bad and can cause breakdowns of essential processes, such as the reproductive process, because they are trying to focus on survival instead of reproducing. Pollutants can affect the physiological function of a fish's HPI and impair the ability to cope with stressors. So the purpose of this study was to determine if variation in riparian land use practices affect the physiology of creek-dwelling fish, using Redfin Shiners as a model species. So to identify Redfin Shiners, the male and females both have a black dot under their dorsal fin. The males are known for their reddish-orange fins. They are found in deep pools and tolerant to moderately poor stream quality. So we hypothesized that variation in riparian land use practices would influence stress physiology of Redfin Shiners. We further hypothesized that there would be difference in water quality accompanying physiological differences. So in order to complete this project, we collected Redfin Shiners from June 9th to August 3rd at 12 different sites using 4 creeks located in Macon County, Illinois. The 3 different habitats that we looked at were agriculture, urban, and protected. We considered it agriculture if it was surrounded by or immediately downstream from cropland. Residential is if it was within the city limits of Decatur, Illinois, and protected if it was a conservation habitat. The 4 creeks that we looked at were Stevens Creek, Friends Creek, Big Creek, and Willow Branch. So this is a map of the city of Decatur, and the dark green line going through here is the Seining River, and the outermost area of Decatur is a lot of protected and agricultural habitats. Most of the protected habitats are downstream from the agricultural habitats. The dots on the map represent the 4 different creeks that we looked at. So the pink dots were Friends Creek, the blue dots represent Stevens Creek, yellow dots are Big Creek, and the orange dots are Willow Branch Creek. The outermost dots are agricultural, and then it goes to protected, and the innermost dots in here are residential within the city limits. So to capture the Redfin Shiners, we used a seining net. We tried to capture at least 20 Redfin Shiners per site, but sometimes we got a few more. Once we captured the Redfin Shiners, we put them into an isolation cup filled with hormone spring water, and we let them swim in there for 30 minutes. As you can see in figure 3, we have a small plastic cup inside of a bigger cup. The small plastic cup has a hole on the bottom of it, so that way when we were removing the fish after the 30 minutes they swam around, We would not contaminate the water. The water would just fall through and go into the bigger cup. Then we would measure the length and sex of each redfin shiner. Now, if we didn't capture all 20 redfin shiners yet at that site, we would snip a piece of their dorsal fin. So that way, when we went back to the site, we would not use the same fish and test their same stress level again. Once we had all of the fish, we would extract their cortisol. We used a syringe with a hydrophobic filter on the end and pull up the water. The cortisol would get trapped in the hydrophobic filter. We would take the hydrophobic filter off and pull methanol through the same syringe. And then we were able to push the cortisol and the methanol into a test tube because the cortisol is soluble in methanol. And then we were able to take the test tubes back to the lab so that way they could evaporate. So the extracted samples were evaporated and the crystallized hormones were reconstituted to 200 microliters of cortisol assay buffer. To determine the cortisol, to measure the cortisol extracted, we used cortisol EA kit. So we put the diluted samples into a plate and the antibodies would grab a hold of the cortisol. And then we put conjugated cortisol from the kit into the plates and it would bind to the rest of the antibodies that had not already binded to the diluted samples. We then added an enzyme and the color would change on the plate. The more blue of the color means the less cortisol that was in each plate. So at each site, we tested the water quality. We did that on the day that we sampled the fish and in the middle and at the end of the study. We determined the stream's pH, hardness, dissolved oxygen, and temperature. We also determined the stream's characteristics, the canopy over and the coarse woody vegetation. So to run our statistics, we used SPSS and we determined it was significant if the p-value was less than 0.05. We ran 3 analyses. The first analysis examined factors influencing fish cortisol levels. The second analysis determined if demographics differed among habitats. And the third analysis assessed variation in stream qualities among habitats. Our results. We sampled 271 redfin shiners with a survival rate of 95.6%. We only had 12 redfin shiners die throughout the whole experiment. It was concluded that the canopy, woody debris, and hardness were not significantly related to the fish cortisol. Figure 6 here shows the average cortisol of the 3 different habitats. The agriculture habitat had significantly higher cortisol than the other 2. In figure 7, we are looking at the demographic variation of redfin shiners compared to the 3 habitats. The agricultural habitats, the juveniles, there was a lower ratio of juveniles compared to male and females. The protected site was pretty evenly distributed, and the urban site, the females were heavily skewed towards. In figure 8, we are comparing the average pH value at each habitat, and the agricultural habitat had the significantly lower average pH than the other 2. In figure 9, we are comparing the average dissolved oxygen level of the 3 habitats. And once again, agriculture had the significantly lower average dissolved oxygen level. So our data did support the hypothesis that different riparian land use affect the physiology of creek-dwelling fish. The redfin shiners in agricultural sites had significantly higher cortisol levels. And in agriculture sites, the pH and dissolved oxygen levels were significantly lower. This can indicate the dissolved oxygen and pH may be key contributors to physiological differences. The age and sex structures among the sites differed. There's a lower ratio of juveniles to males and females in agricultural sites. Protected sites had the best distribution for population growth. Past research has actually shown that the fertility of fish decreased in more acidic water, which this could make sense why there was a lower ratio of juveniles to males and females in agricultural sites, because agricultural sites had a lower average pH than the other two. That this can help indicate that adults may use agriculture habitats for foraging, but rely upon the protected areas for successful reproduction. Past studies have also showed that dissolved oxygen levels affect the fish feeding, growth, and metabolism. There's also a past study, which Dr. Wilcoxen is about to explain in the next study, that fertilizer chemicals can run off into treats and affect the stress physiology and immunity of fish. And there was a study done on species related to the Redfin Shiners, and they responded the same in different temperatures, but their masses increased in the cold. So future research that we could do, there was a study done with treat chubs looking at forest streams to agricultural streams, but the only difference was they used electric shock instead of a seining net, and they still got the same response. But electroshock is actually more stressful to the fish than a seining net would be, which is why we used a seining net. We could also use different species, such as top minnows or darters or suckerfish, and see if the same patterns hold up in other species. We could take a blood sample from the Redfin Shiners, but this would be lethal to the fish. We did have a large sample size, but we could have had more sampling sites. We tried to use Spring Creek, but there were not many access sites. And whenever we did, the water quality was poor and we did not find any Redfin Shiners. We could also try to sample at a different time of the year. We sampled during the summer, but we could try if it's different in the fall or in the spring. And I want to personally say thank you to Travis Wilcoxen for guiding me through this scientific research project. Thank you to Claire Rears for assisting me in collecting some data. I appreciate all the support from the Millikan Biology Department, my family and friends. This work was funded by Billings Research Summer Research Fellowship from Millikan University. Any questions?